This is going to be such a big thing. I'm still amazed by what the cells can do. To date, there are no known severe long-standing effects. The potential in this is huge.
Mattias Bernow is CEO of Cellcolabs, a biotech company making high-quality stem cells available at scale.
Stem cell therapies have the potential to do for chronic diseases what antibiotics did for infectious diseases. Where truly, I believe, in a pivotal point of time, as a doctor, it's so interesting to see, okay, what can be done using the cells?
In this episode, we talk about the future of regenerative medicine and how stem cells could be used to unlock new treatments for chronic illness.
Being CEO of a pharma company, what I really look at is what does science say. If you ever consider going for a stem cell treatment, make sure that the cells are...
Hi, thank you for joining us for this new episode. Today we are talking about stem cells. I imagine a lot of you are curious about what is stem cells and who to look at. So please stay tuned for today's episode, and if you love it, please subscribe to our channel. Your support and your like will make us grow further. Thank you. Hi Mattias, thank you for coming to the HumanWare Project recording.
Thank you so much. Thank you for having me. Happy to be here today.
Could we start right away on what is stem cell?
Stem cells is the original cell type that can differentiate to being other different cells within the body. You have skin, you have kidney, you have heart, you have brain. And everybody knows that it all starts with a sperm meeting an egg, there's a fetus, and then there's a baby, and then we grow up. And from those original cells, they differentiate to become different cells. And there are the original stem cells, and later on there are later stage stem cells that can become not everything, but different things. So a stem cell, the stemness of a cell is how much and for what it can differentiate to become into. The reason in the past 20 years why stem cell research and stem cell treatment has really become a huge thing is due to the potential that they could have in modern medicine. And to position stem cells, I often quote a former dean at the Harvard School of Medicine, Professor Joseph Martin, who said that stem cell therapies have the potential to do for chronic diseases what antibiotics did for infectious diseases.
Okay.
Yeah. So what did he really say? Well, if you think about it, 80 years ago, before Alexander Fleming invented, or rather discovered penicillin, you could cut your hand and die from blood poisoning.
Yes.
Children died young from simple infections. Old and frail people also died from simple infections, and people like you and me, midlife, full health, could die from a simple infection if we were unlucky. That does not happen today, 2025. With the availability of antibiotics, we can treat most infections. Today, instead, we're dying from chronic diseases, such as heart infarction, stroke, Alzheimer. We suffer from diabetes and life-impairing diseases such as arthritis. And for many of those, if not all, we have really no good cure. We can sometimes treat the symptoms, but not really cure them in a good way. So what Professor Martin said was that with the potential of stem cells, there may be a new tool in the toolkit of modern healthcare, where we may be able to alleviate some of this disease burden, maybe even to cure some of them, but even more interestingly, and why I, as a physician and CEO of a pharma company, why I think this is super interesting is that stem cell therapies may have the potential to really prevent some of these chronic diseases. This is going to be such a big thing. Just like there is a before and after antibiotics, in modern healthcare we're now at another pivotal point in the history of medicine where there will be a before and after stem cells. And that is what is happening right now around us.
So we're here today in the Cellcolabs office in Sweden. Could you tell us what type of stem cell you guys are working on?
Here at Cellcolabs, we work on a certain kind of stem cell that is called MSC, mesenchymal stem cell. They have been known for 40 years plus, and they are in huge demand and interest right now due to the things that they can do. When you mostly hear about people going for stem cell treatments, whether it's for a severe disease indication or if it's more from a preventative and longevity perspective, most of these cases they are using MSCs, mesenchymal stem cells. The reason why we are working with this is that we're based on 25 plus years of world-leading research by world-renowned professor Katarina Blanc, who's a professor here at Karolinska Institute in Stockholm. And Cellcolabs were built at Karolinska Business Park. As you can see, the red brick buildings around where we're sitting right now. This is Karolinska. And on the other side of the street, you'll have Karolinska University Hospital. Professor Katarina Blanc, she's a hematologist, so a blood doctor by training. And when she was doing her research work in the late 90s, she realized that for a certain type of patient that she was working with, leukemia, so blood cancer patients, where the treatment is a bone marrow transplant. For some of those cases, the transplant did not work completely, so there was not a perfect match between the donor and the receiver. They then get the disease called graft versus host disease, which is a terrible disease where the bone marrow graft is trying to throw out the patient's body. Severe symptoms and often terminal. That is when Professor Blanc realized that maybe those mesenchymal stem cells can be used to treat that disease. And she was right. Growing large numbers of MSCs, she showed that she could save the lives of 50% of the terminally ill patients in GVHD. She was therefore published showing this in Lancet 2004 and again 2008. And after that, the research field about MSCs just increased and multiplied several times. And after that it became the big thing that MSCs treatment now are. And why is that? And why did Professor Blanc think that this would actually work? Well, at that time there was a lot of hypotheses in the late 90s, but now, 25, 30 years later, there is a common consensus in the research field that MSCs are interesting for several reasons. We know a lot of them. We still don't know all. There's a lot of research still to be done, but there are three overarching things that makes MSC treatment highly interesting. The cells are A, anti-inflammatory, suppressing inflammation. As you know, inflammation is the main driver behind chronic diseases, aging, and several types of cancer. On top of being anti-inflammatory, they're also regenerative and can help heal damaged tissue. For example, if you have a cartilage tear after your morning 5K run or yoga, or if you have skin damage after UV exposure, or if you're in a severe car crash, then you have tissue damage. And the body normally replaces damaged tissue with scarring tissue or fibrosis. But with the MSCs, you can replace some of that and regenerate the cells with functional tissue, which makes them super interesting. So on top of being anti-inflammatory and regenerative, they're also immunomodulatory. A difficult word that means that the cells can modulate your immune system. If it goes rogue, starts attacking yourself as in autoimmune disease, or if, as with increasing age, if it goes weaker and weaker, the MSCs can be helped to boost the immune system. So for these three reasons, the cells are almost interesting for any indication. You may even say that they're sometimes like a solution looking for a problem, which is shown by the current global research. If you look at research now on mesenchymal stem cells, there are at any given time between 400 and 600 ongoing clinical trials using MSCs for different indications. And I often say that if you put up an organogram of a hospital, you can throw a dart on that organogram, and whatever clinic you hit, there's an ongoing trial.
What are the MSCs are good at curing, and what do they count, what they're working with?
If you stick just to the ongoing clinical research, the most currently researched area are actually the neurodegenerative diseases. Diseases where we have no good treatment, and where there's a great hope given the function of the cells that they may actually help. So neurodegenerative disease is number one when you look at the number of ongoing clinical trials. Number two is actually joint disease, especially led by osteoarthritis. Arthritis is the fourth biggest disease burden globally when it comes to people not being able to go to work or live their life due to pain. The third one is cardiovascular disease, a main killer, both with myocardial heart infarctions and strokes. And there has been a lot of research. They all confirm the safety profile of the cells, and they show interesting initial results. And depending on the indication, they have progressed either to phase 1, 2, or 3 trials. And if you ask me, and I would look into the future, I think over the next 5 to 10 years, we're going to see new approved clinical drugs where this will be used. And I think that this will be a new tool for healthcare to start treating and alleviating and even preventing maybe the onset of many of the chronic diseases.
How can the stem cell treatment help with the people who are having diabetes nowadays?
So looking into diabetes, one of the big global diseases that are increasing, whether it is the way we live or for other reasons, current research is looking at if the cells can be used to uphold the child's own insulin production for longer. And early trials show that that may be the case. They confirm the safety profile, and it seems like the patients may be able to uphold their own insulin production for longer, which would be amazing. One of the challenges, and I think we'll get back to that several times during our conversation, one of the challenges with MSCs is that they are quite scarce. There are too few of them in the world, and they are quite expensive. And that's a problem not only for patients, but also for research. Because when the research becomes too costly, it doesn't accelerate at the pace it could. So for example, in many of the diabetes type 1 clinical trials, the patient gets one treatment and it seems to be beneficial. But what if the patient could get several treatments over time? Maybe it would be possible to uphold their own insulin production for longer. The dream when it comes to diabetes type 1 would of course to understand when the disease actually starts. And the problem, we can dive deeper into that, but that is you don't notice it until a couple of weeks later. What if we can find out a way to treat when the disease really starts? Would it be possible to refrain from destroying the insulin producing cells and thereby maybe actually cure diabetes type 1? There's a lot of research that needs to be done, but suddenly there is a hypothetical chance that this can be tried at least. When you think about the diabetes type 2 patients, that is more often, that comes later in life and depends on the way we live, body weight, genetics and several other factors. Published clinical trials, they show that for diabetic type 2 patients, when they receive good MSC treatment, it seems like the overall blood sugar can go down, so it's better controlled, and also that it fluctuates less, which is also good, so that you have a lower and stable blood sugar. These are still early trials, but they seem to be have a very huge potential in going forward. And I think it's worth stopping for a little bit, because there are many producers of MSCs and there are a lot of trials and some are more positive and some are less positive. What is important? What is known? How should you produce MSCs in a good way? Well, first of all, it should always be based on science. Number two, every production of MSCs should be produced according to GMP. And if there's something I want your viewers and listeners to take away from today, if you ever consider going for a stem cell treatment, whether it's within a university hospital or a clinic somewhere, make sure that the cells are produced according to GMP.
Tell us as an audience, if this is the only video they're going to watch to understand what are the safe and security lines, what would that be?
GMP is an acronym that stands for Good Manufacturing Practice. That is the highest and also only gold standard globally for how to produce a pharmaceutical. Whether it's paracetamol, ibuprofen, hormones, chemotherapy, or advanced stem cell therapy, it should always be produced according to GMP. But they are the highest and also only level for use in humans. There are a lot of stem cell producers globally out there that are not adhering to GMP. And you've seen our lab, you know how every little detail is controlled, all the way from donor selection to harvesting of the stem cells to taking them into the approved GMP clean rooms, to taking out the stem cells, the MSCs, out of the bone marrow and then producing them, cryopreserving them in the end. Then they're an off-the-shelf product. Then you want to transport them to where they're going to be used, which you do in cryo shippers, minus 190 degrees Celsius. And when they are used, they are thawed up using a controlled protocol. They are checked for sterility, viability. They're counted that you have the right amount before they are being administered to a patient. whether it is within a clinical trial or if it's a treatment given the framework and what is approved at that site.
So if I were an individual patient, I'm looking for some stem cell or some therapy that could give me this MSCs to inject, what are the questions I should ask?
Number one is GMP, good manufacturing practice. Are the cells produced by an approved GMP producer? And not if they're GMP compliant, meaning that they try to follow a framework, but they should be GMP approved by a third body, national FDA. And don't only ask if they're approved, ask for the certificate. If they're approved, I'd be happy to show you the certificate. Number two, what is the source of the stem cells? And when it comes to MSCs, there are four most common sources, and it is bone marrow, adipose tissue, meaning fat. It can be placenta, or it can be umbilical cord tissue. Being CEO of a pharma company, what I really look at is what does science say? That is what I speak from. And when I say something else that is more anecdotal, I try to truly point that out to make sure that the information is correct and that everybody knows what is known and what is not known. And when it comes to published research and ongoing clinical trials right now, the absolute majority of these trials are using bone marrow-derived mesenchymal stem cells. And if you look at those who are more come from commercial side, they tend to prefer umbilical cord cells. There are an abundance of umbilical cords, meaning that's an easy cell source to source, while researchers, they don't care that much about what is an easy cell source. They care about what is the best cell source to procure your cell from. And when you look at published science, it seems like independent of its bone marrow, adipose tissue, umbilical cord, or placenta, the cells can be equally good if, and only if, they are sourced from an optimal donor and they're produced according to GMP. They can have the same potency if you get it right. There is, though, a difference when it comes to risk profile, because umbilical cord cells, placental cells, and also adipose cells, they may give rise to clotting of the blood, which may, if you're unlucky, lead to a pulmonary embolism, which could be fatal. That must not be a problem, because except for asking for GMP and cell source, you should also make sure that you have a good doctor. And if you have a good doctor, the doctor knows that placental, umbilical cord, and adipose tissue may cause blood clots, and that risk can be offset using heparin or a blood thinner. In the same way, these three cell sources, they may be initially pro-inflammatory before they turn anti-inflammatory. And that means that you are, as a patient, more likely to experience a fever, or if injected locally, swelling and pain. While if you use bone marrow cells, they don't form blood clots in the same way. And they are also anti-inflammatory from the get-go, which means that as a patient, you're less likely to experience a fever or local pain when injected. And why is that? Well, we can dive into the science and the other three sources having more tissue factor that leads to this, but it's also quite easy to explain from a layman perspective, because what is the highest risk for mother and child when giving birth? It's bleeding to death. Therefore, umbilical cord cells and placental cells, they're highly procoagulatory. Even the stem cells, when taking out of that tissue, still have a procoagulatory effect. While if you take the cells from the bone marrow, the safest source, if you don't know it, bone marrow is actually the blood-producing organ. So if you take the stem cells from the bone marrow and you put them back in the bloodstream IV, they're at home.
Okay.
They belong to the same cell source that produces the blood. So when you take bone marrow stem cells, there is no risk of, in the same way, of clotting the blood, and they're anti-inflammatory from the get-go. So what do researchers care about? They care about two things. A, not harming the patient, and B, having the best potential effect. And that is why global research tends to favor bone marrow research over other sources. From who should we take the stem cells? So for your viewers and your listeners, let's start, well, why should I take somebody else's stem cells? Why shouldn't I take my own? If you take your own cells, that is called autologous cells, your own cells. While if you take donor cells, it is called allogeneic cells. And for a very long time, it was a lot of research being done on autologous cells. And actually, Professor Katarina de Blanc, whose research Cellcolabs is built upon, tried to spend, I think, 10 years showing that using your old cells could be more beneficial than using someone else's cells, but failed. Because what is important when you source the cells? There are two main things that sticks out. One, it needs to be a person that is between 18 and 30, because that is when the cells are at their strongest. The second thing is that the cells must come from a donor in perfect health. So there's a huge screening process, and we can get back to that. But if you take cells that are from between 18 and 30, from a donor in full health, that is when you get the most efficacious cells, as shown by research. So then I often get the question, well, I know that if you take cells or tissue from someone else, we need to match genetically. If I get blood from someone, we need to match blood type. If I get a kidney from a donor kidney, we need to match genetically. So how about the MSCs? Well, that is the beauty of MSC treatment or MSC's therapy. Those cells, they don't have the surface marker. So your body doesn't recognize if it's your cells or if it's my cells. Therefore, it is better than using any of our cells to take from an optimal donor. And therefore, for us as a production company, our starting point is finding optimal voluntary altruistic donors who want to donate a very little piece of their bone marrow to be able to produce MSCs.
Is that going to cause any harm for donors?
The body regenerates bone marrow. Within 3 to 6 weeks, the bone marrow grows back, so it doesn't make any harm to the donor whatsoever. And here is another deep dive into the donation, because if you do a bone marrow transplant, you need half a liter to a liter of bone marrow, and that's a very big thing for a leukemia patient treatment. While if you take bone marrow for pharmaceutical production with Cellcolabs, we only need 50 milliliters, so a very small amount. So the donor, having been screened, and we can get back to that, once we harvested a very small amount of bone marrow from them, we recommend them to take it easy for 24 hours, but they can go back to work, or they could go to the gym, even though we don't recommend it. Compared to if you're a bone marrow donor for bone marrow transplant, they need to go on sick leave for 2 weeks, because taking out half a liter is so much and take a huge toll on the body. So then, how do we screen the donors? Well, first of all, we just put up signs, or we say that we're now looking for bone marrow donors for pharmaceutical production for commercial use. Then there's a long line of people lining up saying, understanding that stem cell therapy may be the next big breakthrough in modern medicine, yes, I want to contribute with a small amount of bone marrow. So then those who apply to become bone marrow donors, we screen them thoroughly. We go through the family history. Are there any diseases that are known? Where have they lived? What has been their personal health? Where have they traveled? Any risk behaviors? And if all that pans out well, they come to see our doctor for a physical examination. If that goes well, they go for a blood test, and it's a plethora of blood tests. And if all that comes back good, the doctor signs, this person is approved to become a bone marrow donor for pharmaceutical production. And then the clock starts ticking. So then we have a couple of weeks to take out the bone marrow, which we do from the backside of the hip bone. So not from the back or from the spine, but from the backside of the hip bone. And after taking that out, which takes 45 minutes roughly, it's done in a local anesthesia. So they feel the first needle prick for anesthesia, and then they shouldn't feel anything more. Takes roughly 45 minutes. Then we take the bone marrow here to our clean room facility that you saw earlier this morning. And in the clean rooms, from the bone marrow, the stem cells are taken out of the bone marrow. So it's not full bone marrow that is being used, but the MSCs in the bone marrow. They are then grown in 3 passages and multiplied. So from one donor at the end of production, we get out roughly 200 systemic standard treatments of MSCs that are then cryopreserved in vials, so frozen down to minus 190 degrees. And at that level, the cell clock stops. So they can then be transported anywhere in the world using cryo shippers. So they're transported at the same temperature, minus 150 to minus 190 degrees.
How long is the preservation period?
Well, currently we know that we can preserve them for up to 6 months, but that only depends on that we need more calendar time to show that they can be stored for 1 year, 2 years, maybe even 20 years. It's all about storing them in the right way at the right temperature.
Can we go back a little bit for the placentas? Because within the longevity community, everybody's been saying you need to save your placentas. What will be the procedure for that? Would you happen to know that?
There are stem cell banking companies who serve with that service and can help out. But I think it should always be a discussion between the mother and the doctor, understanding how has the pregnancy been, what are the benefits of saving the umbilical cord or placenta, and what benefits can there be to let it stay on for as long as possible, because there may be beneficial effect for the child being connected to the mother to get as much of the blood and stem cells when giving birth to the child directly. But this can be done. I think many people have done it. I get this question on a daily basis, and there are pros and cons. If you've done it, it's fantastic, because God forbid that your child would ever fall sick. Those cells may be, in some cases, used as starter cells, so you don't have to go look for a donor. Given what we see in research coming out and the scientific progress, I think over time that will not be needed, and we will be able to either use donor cells or in time manufacture the cells that may be needed for that person. But for now, if you have it, it may be very good, but I hope that you will never need to use it.
When we think about preserving your own bone marrow, will that be possible?
It is not a thing. It can be done, but I would say that's kind of a commercial approach to it, which adds little, if no extra value, according to the known science of today. Because as research shows, it seems that allogeneic cells seem to have the best effect when we source the optimal donor. And most of us are not optimal donors. But if, just to play with the thought, say that you are an optimal donor, is it still good to preserve my own bone marrow for later use? It could be, but it would come at a significant cost.
What do you see the future will hold to make this process a lot cheaper?
We're already there. Looking at the global price for GMP-produced MSCs, I think we've cut the price already by 50%. And with our continuous improvements of the production process, we make sure to always have the highest standard, and also continuously to decrease the price and increase output. We're working on a big next step, which would be going from the 2D production that you saw today in our production facility to bioreactor production, which is a 3D production. And that will be a step change. Many parts of the industry are looking for this. We're doing quite well right now. We have done a couple of trials runs during this year that has been successful. And with that, we will increase the output by a further 20 or 40 times at least. And we also expect to be able to cut prices maybe 80, even 90% over the next decade using this.
What do you mean by this 3D process? Is that means it's more robotic or more automatic?
There are actually several things that will improve at the same time. Yes, it will be more automated, which is good. It will also be in a closed system, which is also good for the production and the quality. But it's also a completely different way of growing the cells. Going into that rabbit hole, currently the cells, they grow on a plastic surface. So it's a 2D surface. And when that surface is full, you need to harvest the cells and reseed them at a lower concentration, and they grow again. And each of those times is called a passage. And you can only grow them a certain amount of times, and then they tend to get tired and less efficacious. So that's a limit to how much you can grow. With a bioreactor, you can do this in a much more effective way, whereby adding small beads, you can increase the surface and thereby have them grow without having to kind of harm them through the harvesting process. So that's going to be a huge step change in terms of production technique. which we look forward to launch in the next few years.
Given to knowing so many upsides about this treatment, what are the safeties and things to be considered of?
That's a great question. As a doctor, whenever I meet a patient that has an indication, whether it is heart disease or they come from purely a preventative aspect, or may it even be for a beauty aspect, then whatever treatment I prescribe or use, I always think, what is the risk? What is the safety profile, and what is the potential benefit? And the beauty when it comes to GMP-produced bone marrow MSCs treatment is that to date there are no known severe long-standing side effects. And this you can see, you can look at reviews, meta-reviews with up to 62 clinical trials included, and thousands of patients having been treated with GMP-produced bone marrow and mesenchymal stem cells. To date, there are no known severe side effects. This still feels a bit strange to say, because as a doctor, you know that there are always risks. And of course, if you get an IV, if you get a local injection, punching it through the skin, there's always an risk if you don't get it right. But that can be managed if you have a good doctor and a good sterility. But with the products as such, GMP-produced bone marrow mesenchymal stem cells, to date, there are no known severe long-standing effects. And that is why it's, as a doctor, is so interesting to see this. Okay, knowing that, what can be done using the cells? I often get the question, well, can they give rise to cancer? And I can only respond to that from what do we know, what is shown by science. And to date, there are no known cases of cancer that have arisen from MSC treatment. And then one might say, well, there has only been a couple of thousands, tens of thousands, and the follow-up time is just 20 years from the first ones. Long-term effect, may there be other severe consequences? And given what we know about the cells and how they work, that would be highly unlikely for several reasons. One, it has not occurred yet. Two, and this is important, this is more than most doctors actually know, the cells, whether they're given intravenously or locally, they don't engraft. They don't turn into muscle tissue, brain tissue, or cartilage. What they do, once they're injected inside your body, they start listening to the cells in your body. Where is their injury? Where is their inflammation? How is the immune system doing? And based on that, the live stem cells, they respond to those signals, sending out other signals, enabling your own body's healing function, bringing down the inflammation, restoring damaged tissue with functional tissue rather than fibrosis or scarring tissue, and on top of that, working with the immune system, boosting it if it's weak, or setting it right if it started going rogue in different ways. So they cannot turn into cancers in your body. The cells at the end of the process are cleared from your body. They don't engraft, they are cleared after they have started processes in your body. So once you get them IV, they go around your blood system, they stay in your body for 2 to 10 days. When they speak to your own body and activates their own healing functions. At the end of the process, they are actually stuck in the lungs, where they are engulfed by the macrophages of the immune system, skewing the macrophages from type M1, pro-inflammatory, to type M2, which is one of the ways that the cells actually exert their effect.
You answered many questions I'm about to ask. Thank you for that. How do you utilize modern technology to make yourself a better piece of HumanWare?
One of the biggest bottlenecks in the stem cell industry, whether you're a producer or you're a hospital or something else, is that there are so many charlatans out there making false claims and treating with subpar cells that in best case they don't do anything, but in worst case they actually harm the patient, and that is setting the entire industry back, making even doctors question if MSCs or stem cell treatments can be used on different indications. And it's just to look at the published science. One of my favorite clinical trials, it's an Australian company working with cardiovascular disease. Actually, it was a big phase 3 trial on heart failure, where the cells were injected into the heart muscle, hoping for the heart to move better, to improve a condition called heart failure. And it did slightly, but what was truly interesting with that trial, and this was a big phase 3 trial, 500 participants, 250 placebo, 250 who received the cells. In the cell group, the risk for heart infarction decreased by more than 50%. Actually 63% in the high-risk group. What is even more interesting, in the cell group, also the risk for stroke decreased by more than 50%. I think even 67% in the high-risk group. And those numbers are just amazing. Decreasing the risk for both heart infarction and stroke by more than 50%. To do that, I would need to have the patient take more than 5 pills per day for the rest of their life to have the same similar preventative effect. And this was from one-time treatment with one systemic dose of 100 million cells, followed up over 2.5 years from one treatment. So the potential in this is huge. And the ongoing research will show over time for what indications to use them and for what not to use them.
And when you think about the stem cell treatment, how do I know if I need it?
The easiest way, really, to be super simple is just to have a look in the mirror.
Okay.
and if you look healthy, appropriate to your age, then you have obviously a decent supply of stem cells that are doing the things. As soon as you start aging, that depends on several factors, but one of those things is that also your stem cells are getting fewer with age and they're getting less potent. And that may be the time when you could think about what can I do? Well, there are 5 things that you can do for free. The most important thing for health is sleep. Get sufficient sleep.
Yes.
The second most important is actually having a social setting, friends, having people that you speak to on a regular basis. It could be even just nodding to the cashier at the supermarket is good for health. So sleep, social setting. Number 3 and 4 is exercise and eating right. And number 5 is avoid doing drugs, alcohol and nicotine included. That is a bit challenging because globally we have a culture of consuming some alcohol. It is what it is. It's not the most fun to avoid it, but those 5 things you can do for free. On top of that, there are of course things that are under research, like cold plunges and infrared therapy and different kinds of IVs and so on. Those are maybe still more in the early phases and where a lot of research is still going on and needs to be done. But if you look at stem cell therapies, the knowledge has been around for 25-30 years. The safety is well established if you get it done in the right way, right cell source, produced according to GMP. And then there are initial trials showing highly interesting results, and that is what we as a company, we would like that to accelerate by providing more high quality cells for research to accelerate it really.
But if someone really interested in put some new stem cell in myself, will I be able to reverse aging and feel younger?
There are people talking about anti-aging. Mm-hmm. There is no way of stopping the clock. I would like us to speak about healthy aging, because that is what is truly interesting. The body, can we become 100, 120 years old? Nobody truly knows. If we have good genetics and we live in a good way, there's a higher likelihood that we can have a longer, healthier life. And what drives aging? Well, there is several things, and you all know the hallmarks of aging, but one of the main drivers is actually our underlying low-grade chronic inflammation. It starts from around, well, you have it your entire life, but it becomes significant somewhere after 30, and definitely after 40 it increases. And it has this low-grade wear and tear on your body. If you could decrease that low-grade inflammation, that may be a way to counteract the aging driven by inflammation. And that is one of the many reasons why MSCs has been considered a way to improve your healthy lifespan. Because, as I've said, they're anti-inflammatory and suppresses or pushes down inflammation. And for that reason, they may have a piece to play when it comes to the anti-aging, or as I would prefer to call it, healthy aging process.
And could you tell us a little bit more about your clinic in Bahamas?
Cellcolabs has a daughter company in the Bahamas, Cellcolabs Clinical, that is running clinical trials on 2 indications. One is cardiovascular disease prevention. So it's preventing the onset of cardiovascular disease. So it's a preventative approach, whether it is primary preventative, that you are in full health, or that it's secondary preventative, that you may have already suffered from disease and wants to see if you can stop it from progressing further. And the other clinical trial is on musculoskeletal pain or degeneration. And if you are interested in taking part in those clinical trials to help accelerate research even further, and as part of that get the treatment within the clinical trial, you can reach out to the trials and the clinical advisors, and they would then set up a conversation with you so that you can explain why you want to participate, what is your background, explain what is done within the clinical trial, and then give you personal advice on that. So then I would strongly advise to go to cellcolabsclinical.com and book a conversation. And there you can also get more information and approved materials on the clinical trial.
Let's talk a little bit about company culture and where we are located. Could you tell us a little bit about Cellcolabs culture and where we are based now?
We're sitting here now at our HQ at Karolinska Business Park in Stockholm, in Solna. We have Karolinska Institute on the one side of us and Karolinska University Hospital on the other side of us. And we are building on a scientific heritage of more than 25 years. So we need to live up to that level. And everything that we do is based on the integrity of the science, making sure that we've adhered to protocols and that everything is getting done correctly. The way to get this done and get it done fast is by working with the smart and the best. So on top of being based on the best research in the world, we employ superstars. And when you have a team of superstars, you don't want to hinder them. You want to have them run full speed ahead. So that is why we're working with freedom under responsibility. They know almost always what is the right thing, and if they are uncertain, they are always encouraged to ask. But if for some reason me or a line manager is not available, we trust them. So we're based on trust. We trust them to make the right decision. And 8 or 9 out of 10 times they get it right, meaning that we can produce and run even faster. And the 1 or times out of 10 when we don't get it right, we learn from that as a company together. So we win together and we lose together. And when we fail, we understand what we did wrong, we improve, and we get it right, and we run even faster. So we work with the concept that nothing is impossible. Because if you assume that something is difficult, then it will be very hard for you to solve the problem. If you don't respect when others say this is impossible, you will actually be able to find a solution. Maybe you need to test 100 different things, but there will be a solution. And that is how we, in only 4 years now, in the beginning of our 5th, have been able to build this facility, one of the world's largest facilities producing MSCs according to GMP. We've transferred the entire technology protocol. We've scaled it up more than 100 times. We've GMP certified it. And that is by not accepting that anything is impossible.
Could you tell us a little bit of what are the milestones of the companies? Is there any very difficult time you have to come across? What are the mindset there?
It has all been super difficult. It has all been super difficult. As a former mentor of mine said, Mattias, if it would have been easy, someone else would already have done it. And our unfair advantage is that we are built on the research by Professor Blanc. She's one of the leading researchers in the world when it comes to mesenchymal stem cells. Having that as a starting point, we're 25 years ahead of anyone else who would have started. So even though we're on our 5th year now, I would more like think about that we're on our 30th year right now. And that is why we can do things so fast and why there is so much data behind things. So when we run into a problem, it's not likely going to be the first time. We can look in the papers, what has been done over the past 30 years, learn from that, extract, and have a really strong hypothesis on how to solve it, check it, and then move on much faster.
But Mattias, can you tell us what's your driving force?
Being a clinical physician, I my passion is patients and changing health. And the reason for me working as a CEO of a pharma company instead of working with the individual patient is that while I love working with patients, I sometimes get frustrated that I can only work with the patient in front of me. I want to scale up and make the tools of tomorrow available for the patients of today. And in that, I got the fortunate opportunity to become a CEO of stem cell company Cellcolabs, working with MSCs, deemed by Professor Martin, at the former dean at the Harvard School of Medicine, saying that stem cell therapies have the potential to do for chronic diseases what antibiotics did for infectious diseases. Where truly, I believe, in a pivotal point of time, just like there's a before and after antibiotics, there's going to be a before and after stem cells. And if I can help advance that and make sure that stem cell therapies are available not only to the few selected, but to everyone in need globally, that is not only my line of work, it's also my passion and my hobby, and why I am so high on energy in working with this. Then again, of course, supported by the amazing researchers and the fantastic staff that we're having, pushing every day the boundaries of what is possible and what is known.
Could you bring us back into the first time you're meeting Professor Blanc, and how does this conversation start, and how does the company being formed at the beginning?
So I spent some years as a clinical physician, mainly working with emergency medicine at Karolinska University Hospital. I spent some years working with family medicine, both in Sweden and Norway. I spent some time at Boston Consulting Group as a management consultant, working with healthcare, of course. And then I finally found my way home as an entrepreneur. So I co-founded one of Sweden's largest companies for physical health, telehealth for physical care providers, before I found my way to Cellcolabs. And when I first heard about it, it all sounded too good to be true. So I needed to truly do my DD and understand what is the potential of the cells, what is the risk profile of the cells. I got to meet with Professor Blanc, and 5 years into it, I'm still amazed by what the cells can do in terms of what they can achieve on some indications, on the safety profile of the cells and the potential of the field going forward. I think it's sad that they have been held back for a long time for different reasons, and I'm very proud now to work with one of the finest companies in the field, making the cells available and affordable to fast forward research, accelerate it, and also make it available for patients in need where possible.
And during all these years, you never even once questioned the effectiveness of the stem cell?
I question the effect and the safety profile every day, and I'm so proud to see that again and again and again in clinical trials, in meta reviews, the safety profile is confirmed when it is GMP approved cells, especially done from bone marrow. And I'm happy every time there comes a new trial on any kind of indication, because it expands our knowledge on all the indications where it can do the most benefit, and especially on the chronic diseases and the diseases where we have no good cure in healthcare today. I think the promise of MSC treatment is highly interesting. So that is why we need to push and drive research even harder to learn.
When we think about if it's a med school student, they want to consider to work in this industry as a job, what are your advice for them?
Open up your minds to the possibilities of MSC treatment. We'll be providing you with a recommended reading list. Start reading up and understanding, because due to all the controversies that there has been in the stem cell field, many doctors are still not that well educated, and there is a lot of misbelief and misunderstandings. Read up. It is not in the textbooks yet. You need to read the articles, but listen to the conferences and the educations that you can find. Make sure that they are validated in terms of the scientific background. And if you're interested in, reach out to us, and we'd be happy to tell you more and point in the direction where you could learn more. And I think overall, globally, regenerative medicine is increasing, and it's going to be part not only of the specialist care for Alzheimer or heart disease, it's also going to be part of family medicine, taking a preventative approach and taking care of diseases before they hit and establish.
How big is current market cap of stem cell therapy and treatment are, and where you see the future is going for this type of therapy?
As an investor, the numbers look almost too good to be true. But just looking at the stem cell research market, those are counted in the billions of dollars per year. If you look at ongoing stem cell tourism industry, which is a huge field, often in the gray, and where there is now coming out better and better regulations around the world, that is also a multi-billion-dollar market. But where we're truly aiming for and where we're heading to is the approved registered products over time, or with the improved regulatory framework, so with the more allowing use of the stem cells. The numbers, there are so many zeros behind it, it's not even worth mentioning. What if, for example, we're just talking about osteoarthritis, arthritis being the fourth biggest disease burden globally, where we have no good treatment. You can do the anti-inflammatory, you can do the cortisone injection, but over time you will need a knee prosthetic, for example, which only lasts for 10 to 15, maybe 20 years, and then you need to redo it. The cost of arthritis alone is a mind-blowing number. I talked earlier about the potential preventatory or secondary prevention effect for cardiovascular disease. Cardiovascular disease is one of the main killers of our time and age. And then we have all the neurodegenerative diseases. The addressable market for those indications is huge. So I encourage competition, and it's going to be a truly interesting field over the next decade.
Do you guys have any competitors at this stage?
We're happy to have some competitors, but I would rather, those who may be deemed competitors, I would rather call them colleagues in the field. Those ones that I highly respect are doing good research, working with GMP approved cells. I salute everything that they do, they're great. And then we have a lot of lesser competitors or charlatans not working according to GMP, or not following approved clinical trials, or not being approved for the treatments they're conducting. Those are the ones that are risking the field and holding it back to some extent. But that is a challenge, and I think that is why it's good that regulatory authorities, they look at the field and regulate it in a smart way. And globally, currently, there is now a global trend towards opening up the use of MSCs even further. It was started in the 2012 Nobel Prize for the Yamanaka factor, where Japan realized that it was kind of over-regulated and very difficult to do, so they opened up for conditional approvals, which was a way of financing the phase 3 trials really. Korea followed. Then there has been the patient-funded clinical trials, which is also a mean of financing the research. And then there are now accelerated clinical trials, and US is now actually leading the charge. After having been behind for a long time, they're now leading the charge, where they have set up, not on the national level, but on state level. There are states that are now looking at what indications should MSCs be generally allowed for, where there is discussion on arthritis, wound healing, pain management. So there's a global trend right now to regulating the cells in a more allowing way, but always making sure that the cells are produced according to GMP, so the highest quality.
Mattias, as a CEO and as a founder of such a big biotech company, what does your daily routine look like?
That's a great question. So on my personal daily agenda as a CEO, I'm a father of two, and my wife, she's also having a career on her own. So I'm responsible for taking care of the children in the morning, making sure that they get dressed, fed, and that they go to kindergarten and now school. Our eldest just started school 2 days ago. So I often come in around 9 to the office, and then it's a full day of work, always with a business lunch or lunch meetings until 6 o'clock in the evening, and then there's family time, 2.5 hours until the kids go to bed, and then I continue working until going to bed, and then the day starts again. So with this packed agenda, I try to do physical exercise every day in the morning. I do my push-ups, my pull-ups, my sit-ups, and try to exercise and run 2 or 3 times per week. I ride my bike to work every day. That's a great way of getting 40 minutes of biking each day, which you can do in a fantastic city like Stockholm. But not trying to have too high an ambition, which could stop you from getting started in the first place. Yeah. The most difficult things with going out running is actually tying your shoes. Once you've done that, you're good to go. And you don't need to go for an hour or 30 minutes. You can go 10, 15, 20 minutes. Just getting out is the most important thing.
And last question, as a CEO of a biotech company, what do you see in the horizon of biotech or longevity you are excited about?
To me, the truly most interesting thing right now is preventative medicine and also being able to work with the chronic diseases. So for that cause, I think MSCs is a perfectly positioned tool in the toolbox of healthcare. So I'm very happy to get to work with MSCs, given their anti-inflammatory effect, their regenerative effect, and their immunomodulatory effect, and the fact that they may have a benefit on several different indications and the main drivers of disease and chronic disease of today.
Oh, thank you for your time, Mattias. Really enjoyed our conversation.
Thank you so much for having me. Thank you.
Thank you for watching this new episode of HumanWare Project. So that we can bring more experts like Mattias to you, please like and subscribe. Your support means the world to us.